So I'd like to take this opportunity to thank the people who have accepted to come here, and we had a very good sessions for this for these 2 days. I really enjoyed it a lot. I hope you enjoyed it just as much as I did, so far at least. And after this talk, I mean, we'll have lunch, and then we shall reconvene here at 2 p.m. Try to be on time, please. So I will tell you about largely this mode of signal transduction that my lab has discovered something like 20 years ago, more or less, even though things have been evolving since, and which is based on the transduction of transcription factors of the homeoprotein family. And in fact, this mechanism is not totally explained, how the proteins are secreted and internalized, even though we have some elements that might explain how it happens. And some of the part of the explanation is in the structure of the homeodomain, which is the DNA binding domain of these transcription factors of the homeoprotein family, with 2 sequences. A sequence in the 3rd helix of the homeodomain, which is the internalization sequence, and a sequence which spans the 2nd helix and the 3rd helix plus the beta turn between the 2 helices, which is the secretion sequence. Now, based on this internalization and secretion, I mean, we have speculated that these transcription factors could be direct signaling molecules, so to say from nucleus to nucleus, and that they might also regulate translation in the postsynaptic cells. And I don't know why, when we wrote this review in 2000, we speculated that they were regulating translation as well, because we had absolutely no hint that this was happening. And I think it was because of the bicoid case. As you know, bicoid is the transcription factor of the homeoprotein family, which in flies regulates the translation of caudal. And I guess this is the only reason why. But I had put a question mark, so I'll be quite cautious anyway. Now, as you can see here, I mean, the sequences to get in and out are in the homeodomain, and this was a bit of a difficulty for us, because it meant that we could not mutate these sequences without modifying the cell autonomous properties of these transcription factors. And it was almost impossible, I think impossible in fact, to do a knock-in experiment replacing the transcription factor gene by a gene encoding a transcription factor with a cell autonomous only activity to distinguish between cell autonomous and non-cell autonomous functions of these transcription factors. And this is why, I mean, more recently, we have developed a new approach which consists in constructing single-chain antibodies, mini-gene encoding single-chain antibodies that could be secreted by the cells and block the homeoprotein only outside of the cell, thus blocking the transduction mechanism and the signaling properties of these transcription factors while leaving intact the cell autonomous transcriptional activity. And I will go back to that in a moment. Now, this signaling pathway is not something which is really recent, to be honest. It had been shown in plants by the groups of Lucas and Jackson. I will not develop that. It was shown in flies, in fishes, in frogs, chicks, and in mice. So it's a very old signaling pathway, which has been, I would say, developed or invented by evolution before the separation between plants and animals. And if you look at the presence of transcription factors of this family during evolution, you will see that all multicellular organisms and even single cells like Chlamydomonas, for example, I mean, express these transcription, or yeast express these transcription factors, and that we speculate, or I speculate, that this signaling pathway may be a very ancient signaling pathway that has preceded the invention of the classical signaling pathways. And I will go back to that during my talk. Now, I will just start with the flies. And this work was done in the Collège de France there are kids in colleges. So, by Alain Joliot and Michel Volovich here, and in collaboration with the laboratory of Laurence Machat. And I show this fly work with only one slide to bring forward a concept which I think is very important. Now, this is the signaling path. This is what we call the anterior cross vein in the fly wing disc. This is the posterior cross vein. And as you can see here, NGREL is expressed up to the middle of the anterior cross vein. Now, just to be extremely rapid, I mean, we can have the antibody against NGREL secreted by the DPP cells that do not make NGREL. Okay, so we cannot be in the inhibition of NGREL within these cells. The antibody is secreted and blocks the NGREL protein, which is secreted by the patch hand domain, which is here. And when you do that, what happens that you block the formation of the anterior cross vein, demonstrating that you need the signaling with NGREL to have the formation of this anterior cross vein. Now, what you should look at on these slides is the red staining here and the absence of staining here, showing that DPP signaling is blocked by the blocking of NGREL signaling, so that in fact what you have is a co-signaling between NGREL and DPP. Now, if we elaborate a bit further on that, I think it's a very general phenomenon that these transcription factors do not signal by themselves, but signal with partners, and that in many of the experiments that have been done by the community, they either work in this zone by loss of function of the classical signaling pathway or classical agonist, and without the classical agonist, there is no way that you will see any signaling, or they have worked by gain-of-function experiments when you have so much of the classical signaling pathway that you don't need the help of the homeoprotein anymore. And in fact, what we think is that homeoprotein signaling is important when you use physiological concentration of the normal morphogenes. And at this point, I think that nobody knows exactly what are the physiological concentrations of morphogenes in vivo. Frogs and chicks can do it. This work was done in my lab by Marine Bordelais, Isabelle Brunet, Alain Joliot participated, he has his own lab, and there are many other people who have now left the lab, and always my old friend Michel Volovich, who has always been working with me. It was a collaboration indeed with Christine Holt in Cambridge and her group, and some work was done with the Céline Hatton and Jean Thomas, which I will not present today. And indeed, Andrea Wiesenman was extremely important in this story. And at the time she was at the Max Planck Institute with Wolfgang Wurst, and now she's moved to Tübingen. So, a long time ago, I mean, when we discovered that these transcription factors could be transferred between cells, we also knew that these transcription factors were also molecules that were endowed with positional information. And you see here the very clear example that if you mutate antinapedia, which is one of these transcription factors, the cells that normally develop into the antenna will now develop into a leg because they don't interpret their position well. They think that they are on the second thoracic fragment of the fly. So that we speculated, or I speculated, I have a tendency to speculate a little, that if these transcription factors were captured by the growth cones, the growth cone would know its position and then could answer appropriately to its position. But I forgot about this idea because at the time I realized that the transcription factor would have to go back to the nucleus and that something would happen there and the signal would have to go in the other direction, and this would be very lengthy. This is when I went to a meeting at King's College. It was the inaugural meeting of King's College, and I discussed with Christine Holt, who had just discovered that guidance involved local translation in growth cones. And remembering that bicoid can regulate the translation of caudal, I speculated that these transcription factors may act locally in the growth cone by regulating translation, and I proposed a collaboration to Christine, who was kind enough to accept it. What you see here is the classical guidance paradigm, where the nasal temporal axis of the retina will project on the posterior anterior axis of the tectum. And this is due to the graded expression of FA along the temporal nasal axis and the graded expression of F A along the posterior anterior axis of the tectum. So that when axons from the most temporal part of the retina reach the posterior tectum, they collapse because ephrine will produce the collapse by interacting with the FA receptors. It happens that NGREL regulates ephrine transcription. It also happens that NGREL is expressed in the tectum, that its expression is graded along the anterior-posterior axis. Not only NGREL is expressed in the tectum, but it is extracellular. 5% of the protein is outside, and the outside NGREL also shows a graded expression, so that we could speculate that NGREL might participate in axon guidance. And to summarize what we did, we expressed single-chain antibodies against NGREL in the posterior part of the tectum. And when you do that, you can see that the red guys, temporal axons, that normally will not invade the posterior part of the tectum, will now be able to travel to the extreme posterior part of the tectum. So we don't see the breaks anymore. And this is also true in the cheeks. If you electroporate the antibody in the cheeks, you see that some temporal axons will project to the posterior part of the tectum, which did not happen with other single-chain antibodies. I will not elaborate on that. The mechanisms indeed are a bit surprising. You see here the Mu-Ming-Pu turning assay, where we have the temporal axons and nasal axons navigating in the gradient of NGREL. This is a blue gradient of NGREL. And you can see that the temporal axons are repelled and the nasal axons are attracted in such a gradient. This requires NGREL internalization. A non-internalized mutant doesn't show the guidance effect, and you need also to have a full-length transcription factor. If you mutate the N-terminus, you remove the N-terminus of the protein, you don't have any more the guidance, even though the protein is internalized because the homeodomain is intact. So the big surprise came when we analyzed whether this was dependent. It was not such a big surprise, to be honest, dependent on transcription or translation, but we could verify that, transcription inhibitors had no effect on guidance, and in contrast, the translation inhibitors had a big effect on guidance. Also, if you cut the growth cones off the shafts of the axons, you still have guidance, showing that this is really a local translation effect, which is triggered by a NGREL. It goes through the mTOR pathway. I don't go into details. You can read it in the good literature. We cannot forget that ephrines also do it. So what we did at the time was to make stripes of ephrine, high concentration of ephrines, and we could verify, in fact, that the temporal axons avoid the high concentrations of ephrine, thus forming bands, as you can see here. Now you can go down to 0.5 microgram per ml of ephrine, and you still see the bands. And if you go to 0.1 microgram, now you have a stochastic growth of your temporal axons. But if you add NGREL into the bath, you restore the formation of bands. And again, what you see here is that you have an interaction between NGREL signaling and ephrine signaling, as we had between NGREL and DPP in the flywing disc. You can see it also by the growth cone collapse assay, and which was developed in the lab by Stettler and Ken Moya, Olivier Stettler and Kenneth Moya. And you can see here that you can have high ephrine concentration that triggers the collapse of your growth cones, but you have sub-threshold ephrine concentration in which you have no collapse, except if you add NGREL in the DAF, where you restore collapse. And the restoration of collapse is not obtained with the internalization mutant, is not obtained if you block NGREL internalization, is obtained when you inhibit transcription, but is lost when you inhibit translation. So you have this signaling that you can monitor by this assay, very simply. We had to identify, indeed, which messenger RNAs are translated in the axons, in the growth cones when you do that. And I will not develop very much, but I will summarize that 60% of the messenger RNAs that we have identified in the growth cones are RNAs that encode mitochondrial proteins. And the idea is that the protein gets in, regulates the translation of messenger RNA encoding mitochondrial proteins. The proteins are internalized by the mitochondria, and this is followed by ATP formation. This work was done by Olivier Stettler primarily, and Kenneth Moria, and François Cassagnier. Now, you can see it here. This is a pretty rapid mechanism. This is minus NGREL. This is the ATP 100 second later when you add NGREL to your growth cones. And the mechanism is been published, but you have the secretion of ATP, the degradation of ATP into adenosine, the binding of adenosine to the A1 of the adenosine 1 receptor 1, excuse me. And then this is what, in fact, interacts with different signaling. So that, in fact, what you have is a ménage à trois mechanism where you need NGREL, and then you involve the adenosine R1 receptor signaling, which interacts with the different signaling. So it's much more complicated than we thought at the beginning. So this is a review that we wrote in 2003, I think, with Alain Joyot, where we had proposed that this transfer of transcription factors between the growth cones and the target might lead to the axon guidance, and I think this is more or less demonstrated in the case of the retinal ganglion cells. And we also had speculated that, in fact, this exchange, including local translation, may have something to do in the interaction between presynaptic axons and postsynaptic elements. And if I show these slides, it's just because of the beautiful work shown yesterday by our friend Arjen Schumann. Now, mice can do it. And this is a work which, as you know, it has been alluded to a lot this morning. So it was done in my lab by a lot of people, including, indeed, Clémence Bernard, who is still working here, Eva Bertini, Marine Bordelais, who is left now, Ariel Dinardo, who is really, I mean, the sort of, you know, he has a brain on this topic here. Anthony Perrault has left, Julien Spataza has left, Clémentine is still here, and Michel, I mean, as usual, I mean, is doing a lot of work with us. It was a collaboration with the lab of Takao Hench, and the first experiments were done by Sayaka Sugiyama, and now Hing-Chung Lee is working on the project. And we got some mice from Dario Campora and Antonio Simeone, and also from the lab of Isao Matsuo. So I don't have to develop what the critical period is at this point. I mean, you know it by now, I guess. But what we saw in 2008 is that parallel to the maturation of the PV cells, that you can see by the increased expression of parvalbumin in these cells, we have an increased expression. We said expression at the time of OTX2 in these cells. Now, the problem is that OTX2 is not synthesized in the cells if you use the in situ hybridization or OTX2 GFP mouse of Dario and Campora. You still you see no expression, even though you have the protein. If you do the RT-PCR, you do see no expression in the visual cortex. And if you recombine the OTX2 locus using a driver like CAM kinase 2, which is not expressed in PV cells, you lose OTX2, showing that, in fact, there is some OTX2 done somewhere, not in PV cells, but which accumulates in PV cells. And if you recombine the locus, you lose the PV maturation. So there's a link between PV maturation and OTX2 accumulation. Now, this is why with Takao, and I'm going fast because this has been published, we decided to infuse OTX2 in the cortex before the opening of the critical period, and we could verify that OTX2 forces the maturation of the PV cells. This is the infused side, the non-infused sides, and much to our surprise, I must say, I went to the Riken at the time and I could not believe it. It is specifically captured by the PV cells, which was very surprising. I won't tell you why it was surprising, but it was surprising. It meant that these PV cells may express binding sites for OTX2. Now, when we did that, in fact, we could open critical plasticity earlier in time at P18, when it is normally closed, And it was also closed earlier. So the entire process of maturation is accelerated when you infuse a TX2, okay? When you remove one allele of a TX2, you don't open the critical period, at least at the stages where we have looked. Now, the big surprise was that, in fact, the TX2 was specifically captured by the PV cells when it was infused in the cortex. And we speculated with Takao at the time that the perineuronal nets might be responsible for the specific recognition of a TX2. PNNs have been described by Golgi something like 100 years ago. Carral didn't believe it. He thought it was a fixation artifact, but we were not properly friends, I think. Still, we had to sort of demonstrate that, and we used the Cantonese ABC treatment that you know. Now you remove the PNNs stained by the selectin WFA, and when you do that, you can see that the endogenous levels of a TX2 in these cells is much reduced, leading us to identify in a TX2 a sequence which is responsible for the binding to the cells in the cortex. This sequence has been already shown by Takao, and what you can see here is that you remove the sugars, this protein here doesn't bind anymore. So we had a good hint that this sequence is responsible for the specific recognition of the PNNs by a TX2. You saw this slide already. The highest affinity of this peptide for complex sugars is for CSC and CSD, suggesting that you need two sulfates. And since Iguri, has shown that it could be the balance between this one and this one is very important for the specific recognition of a TX2 by PV cells. I don't go into we infuse the TX2 with the AA mutation in the cortex, and what you can see here is that it can still be internalized by PV cells, but it is now also internalized by other cell types, leading to a dilution of a TX2 inside the cortex. And we developed an OTX2 mouse now, in which we did the knock-in of AA into the RK, replacing, this is unpublished, replacing this RK by a doublet of alanine. You can see here the wild-type animal. The heterozygote, you can see a lot of cell of OTX2 is now lost between the PV cells. And the result of this dilution of the TX2 is that, in fact, you have more cells that are stained with the antibody from wild type to the homozygote and heterozygote and homozygote, but also that this accumulation of a TX2 that you see with time in wild type PV cells is retarded in the mutants, so that it takes longer probably to get the appropriate concentration of protein in the cells to open plasticity and possibly to close it. What Clémence, this is the work of Clémence Bernard in the lab, has shown is that, in fact, if you look at PV staining or WFA binding to PV cells, the point where you have a maximum, the maximum of expression or assembly of PNNs or expression of PV is now P100. P100, according to the work done in Takao's lab that I am not showing, is the time when the heterozygote opens plasticity, in fact. So that it's seen that plasticity opens later in the mutant because the cells mature slower because the TX2 is not internalized as fast as it should be. And we have used this trick to start doing RNA sequencing on PV cells and looking at genes which would be regulated differently at P30 and P100 between the three mutants. One is the wild type, the AA heterozygote, and the plus GFP. And you can see here that this pattern indicates that these genes may have something to do with plasticity. ARC is not surprising, FOS is not surprising, and we have 50 other candidates that show the same pattern of expression are good plasticity candidates, and I will not tell you what they are at this point. Unless you want me to do it. This slide you've seen already. Takao has shown you that if you infuse the RK peptide, you sort of rejuvenate your cells. I mean, in terms of a TX2 content, PNN assembly, and PV expression, and that this can reopen plasticity, that you can take a normal mouse, infuse the peptide, and make it blind in the adult, which is not extremely nice, but you can do reverse. You can take a mouse which was blinded during the critical period, thus has developed amblyopia, and you can now restore visual acuity in the adult by infusing the peptide and letting the mouse run. So this was nice, but at the time when we published the paper with Takao in 2008, we were asked where the protein is coming from, you know, and a very good target, I mean, source of OTX2 was the eye. So what we did was to inject OTX2 in the eye and to show that OTX2 was transported to PV cells. So OTX2 can do it and probably does it, okay? But for some reasons that you will understand very rapidly, in fact, the reason is that OTX2 is not only present in the visual cortex, but it's present throughout the cortex. We are not fully satisfied with this source only. And we were looking for another source of OTX2, and this other source of OTX2 had to be a general source. This is why we were intrigued and excited, in fact, by the fact that OTX2 is heavily expressed in the choroid plexus. You see it by in-site hybridization. This is the OTX2 GFP mouse. This is an antibody against OTX2 in the choroid plexus. And the choroid plexus is called the kidney of the brain. This is not very nice, okay? The choroid plexus is a very important structure that secretes a lot of good factors for, well, you know, you can live without a brain, not without kidneys. So kidneys are pretty important, maybe more important than brains for some persons. But so this is a very, very important structure. I mean, physiologically, I mean, people start realizing that it's an important structure. So what we decided to do, and Julien Spataza did most of this work in my lab, was to specifically recombine OTX2 in the choroid plexus. And when you do that, what you have is decrease of expression at the messenger RNA and protein level in the choroid plexus, not in the retina. And you guess, I guess, what we could show is that when you recombine, now you decrease the OTX2 content in PV cells to the level that you have the onset of the critical period, and that you reopen plasticity. So this is the experiments. You have less OTX2 in PV cells. You have less PV maturation. PNNs do not assemble as they should. And as a result, you can induce amblyopia in an adult mouse, a not nice experiment, but you can also cure the adult mouse from amblyopia, which is a nicer experiment to do, even though we kill the mice after anyway. So that we have this result, which is that OTX2 is synthesized by the choroid plexus, is transported to PV cells, recognizes the proteoglycans or glycosaminoglycans, CSD, CSE, we don't know yet, at the surface of the PV cells, is internalized by the PV cells and does its job. This is very reminding of the French flag problem, because I always thought that these transcription factors are morphogenes. So I always had this French flag problem, well, it was named French flag problem by Louis Volpert, who is an English, okay? I'm not responsible for that. And the idea is that you have thresholds, so that at a certain concentration after diffusion of the morphogene, all cells will turn blue. At intermediate concentration, they will turn white, and at the lower concentrations, they will turn red. That's the French flag problem. And this can only work because you have thresholds at which the concentration will modify the fate of your cells. Now, this is the same thing, except it is a temporal flag. OTX2 is internalized by the PV cells, and up to a certain concentration, plasticity is closed. You pass the first threshold, plasticity opens, you pass the second threshold, plasticity closes again. So you have blue, white, red, but this is within one cell type, and this is with time and not with space, okay? The nice thing is that if you can pass the second threshold back, then you can reopen plasticity. And that is what we've done using the RK peptide and recombining OTX2 in the choroid plexus. As I said, I mean, as you may have anticipated from the talk of Takao, in fact, many PV cells throughout the cortex, auditory cortex, basolateral medulla, somatosensory cortex, visual cortex indeed, have OTX2 in their PV cells, suggesting that this mechanism might not be restricted to the visual cortex, that OTX2 might be a more general regulator of plasticity in the cortex and a coordinator of plasticity in the cortex. And if you look at these little GABA glutamate oscillators in different regions of the cortex, indeed they are modified by the classical neuromediators that we know work in the brain and are very important for the regulation of these oscillations. But we believe that the OTX2 coming from the choroid plexus might sort of coordinate the activity of these different oscillators and thus participate in cognition at large. So just to summarize, sort of, what have we shown? We've shown that these transcription factors can be recognized by specific sugars. So one question is there a sugar code? Well, we think there is a sugar code, that OTX2, when infused in the cortex, is internalized by PV cells, but if you take Engrel, this does not happen. So this means that these different transcription factors may be recognized by different sugars. And this is a work which has been initiated in the lab by Clémence, and it will be pursued in the context of two collaborations, in fact. Now, then it is internalized for the mechanism that we have not totally understood yet, but within the cells, it will regulate translation, and we have to identify all the translation targets. Many of them are mitochondrial targets, meaning that when the homeoprotein is internalized, at least Engrel, but very possibly OTX2, you increase ATP formation. So you activate the supply, you increase the supply in energy of your cells. And this is important, in particular in the PV cells, which are very high and very important energy consumers because of their frequency of oscillations, 80 Hz. This is not, you have to depolarize and repolarize 80 times per second, meaning you have to pump the ions inside the cells, which requires a lot of ATP because you need the sodium-potassium ATPase. Now, we don't know yet whether OTX2 does it, but we're looking at that. The other translation targets have to be identified. We have some of them. Do we always need a co-signaling? I think so. We've shown it for PAC6 in the spinal cord. I had no time to show that. We showed it for Engrel in the wing disc. We showed it for Engrel in the tectum. So we think we always need a co-signaling, but it cannot be a general rule based on three examples. And then it goes to the nucleus. And then it regulates transcription, and we have to identify the transcription targets. As you could see, we have started to do that, and also the epigenetic targets. I will not discuss the epigenetic targets, but we think that these transcription factors can modify the epigenetic status of the cells after they have been internalized, okay? I would like to draw your attention to the fact that there are more than 200 homeoproteins in the vertebrates, and that most of them have the sequences which are necessary to get in and to get out. We've tried between 10 and 13 in vitro. They all do it. Only 4 of them have been tried in vivo, so I will not say that they do it in vivo as well. But if I had to bet, I would bet that most of these transcription factors are in fact signaling molecules. So this is more or less the last slide. But what I would like to insist on is that OTX2 is not the only actor in PV cells, since we know that there are many factors, and we heard about a few more of them this morning, that may regulate plasticity in the cortex. But it could be that it is a very essential factor because it might be responsible for the assembly of the PNNs and thus for the ability of the cells to recognize the other factors that participate in the regulation of plasticity. So just to end, I mean, this work has been funded and supported by many sources, I must say, including, I mean, the national agencies and the European agencies. In particular, we had one grant with Takao at the Human Frontier Scientific Program, also by the industry and our doctoral school, University Pierre et Marie Curie. Indeed, Collège de France, INSERM, and CNRS have been extremely important to keep this work going for so many years. Thank you.